A very warm welcome to all of you guys. Welcome back in another video tutorial for microbiology with Shagnik. So today we will going to talk about or we will going to start a new video lecture series that is on enzymatic reaction. In that particular video lecture series we will going to talk about the mechanism of action of some important enzyme. So in the first video of this video lecture series we will going to talk about chymotrypsin. So let's get started with the video, friends. So this chymotrypsin is a enzyme which is mainly synthesized in the form of its precursor in the pancreas. Okay, and its precursor is chymotrypsinogen, which is obviously metabolically or enzymatically inactive. Okay, so chymotrypsinogen. Now this chymotrypsinogen will be cleaved and converted into active chymotrypsin. Okay, now this active chymotrypsin is nothing but a 3 polypeptide interconnected via disulfide bond. Okay, and if you have watched my peptidase video on protein lecture series, then you know that this chymotrypsin is an example of endopeptidase, right? And it generally cleaves after phenylalanine, tyrosine, tryptophan, and one amino acid that I have not mentioned in that particular video, that is methionine. After this 4 amino acid, the chymotrypsin will work, right? It will hydrolyze the peptide bond. Okay, along with that it will also hydrolyze ester bonds. Okay, because when we will going to talk about its mechanism, you will find that chymotrypsin needs a carbonyl group to work. Okay, so here ester bond also have carbonyl groups, so chymotrypsin can also act on ester. Okay, so that is what I have written here. So here I have all thing I have mentioned, right? So let's take a screenshot to use this for your further study. So now we will going to talk about its mechanism. Now this chymotrypsin is an example of serine protease, and the main important feature of this serine protease group of protein is these proteolytic enzymes, because they are obviously proteolytic enzymes as its name suggests, they are protease. So these proteolytic enzymes mainly act due to the presence of highly reactive serine residue at its active site. Okay, that means serine protease group of proteins have highly reactive serine residue at its active site, which is mainly responsible for its catalytic mechanism. Okay, in case of chymotrypsin, this serine is present at the 195 position. That is why we are calling this type of serine as serine 195. Okay, so what will happen here? Here the C double bond O group of the peptide will be attacked by a very powerful nucleophile. Okay, that is the idea, and that's how serine residue present in the active site of the serine protease will going to covalently bonded with the substrate and form enzyme substrate complex. Okay, so let's see what I have told you. So chymotrypsin is a serine protease, and that's how it works. So another important examples of serine protease are trypsin, then elastase, subtilisin, thrombin, acetylcholine esterase, and other related enzyme. So this type of protease can be blocked by organofluorophosphate such as diisopropyl fluoridate. Okay, so this diisopropyl fluoridate will going to act with the serine 195 in the active site of the enzyme, and that's how they irreversibly block the activity of chymotrypsin. And this simple observation suggests us that the presence of serine 195 is very important for chymotrypsin, not only chymotrypsin but any kind of serine protease to work. Okay, so this serine protease also have a catalytic triad. This catalytic triad consists of serine that we have already mentioned, Along with that, histidine, which is present at the 57 position, that is why it is known as histidine 57, and aspartic acid, which is present at the 102 position, that is why aspartic acid 102. Okay, and they will together create a energy relay system, which we will call as catalytic triad. Okay, and it is very, very important. So this catalytic triad will act as a charge relay system. So with the help of a diagram, we will going to understand how this catalytic triad is formed. So this is the catalytic triad. As you can see, aspartic acid 102, histidine 57, serine 195. So we all know that serine has a OH group as a side chain. So the H will form hydrogen bond with the imidazole group of the histidine. Similarly, another NH present in the imidazole group of the histidine will involved in hydrogen bonding with the carboxylate group of the aspartate. That's how the catalytic triad or our energy relay system is formed. Now what happened during the action of chymotrypsin, the oxygen which is present at the side chain of serine will going to react with the C double bond O present at the peptide. Okay, that's how they covalently bonded with the enzyme and form enzyme substrate complex. Okay, and during that time this histidine will going to act as a general base, and it will going to abstract the serine proton. Okay, and aspartate which have negatively charged, this aspartate will going to stabilize the positive charge on histidine. That's how the entire thing is going on in case of the hydrolysis of the peptide bond with the help of chymotrypsin. Okay, so that's the whole thing. So for a quick recap, this chymotrypsin is a serine protease group of enzyme, and serine protease mainly works due to the presence of the serine residue at its active site. Okay, this serine residue will going to react with the C double bond O group of the peptide or ester. Okay, and that's how they form enzyme substrate complex, and then the entire thing is going on, right? And here the catalytic triad is important, that is aspartic acid which is present at the 102 position, that is why aspartic acid 102, histidine 57, and our serine 195. Okay, this serine 195 will be hydrogen bonded with the imidazole of the histidine, and this histidine will be hydrogen bonded with the carboxyl group of the aspartate. That's how the entire catalytic triad or energy relay system is formed. Okay, and when the reaction is going on, the oxygen which is present at the side chain of the serine will going to react with the carbonyl group of the peptide, and during that time this histidine will going to start acting as a general base, and it will going to abstract the serine proton. Okay, and the positive charge which is now on histidine will be stabilized by this aspartate, and that's how the entire thing is going on in case of chymotrypsin. Not only chymotrypsin, but in every proteolytic enzyme which is under the serine protease group. For example, your trypsin, for example, your subtilisin, for example, your elastin, for example, your acetylcholinesterase, and blah blah blah. So here another important MCQ is the organ name of the organofluorophosphate, which can be used to block the activity of the chymotrypsin, that is our diisopropyl fluorophosphate, right? Sorry, diisopropyl phosphofluoridate. So that is the idea of our how the chymotrypsin works. That is this one, diisopropyl phosphofluoridate. So that is the idea of how our chymotrypsin works. So I hope this video will be helpful for you. Thank you.